Hi, today we will be presenting about epidemiology and diagnostic technique of Enterobius vermicularis infection. First thing first, let me introduce our team. We have DALELA and me as ISMA, AQIL and UMME. Let's take a look about life cycle of Enterobius vermicularis. The life cycle of Enterobius vermicularis is simple and is complete in a single host, and no intermediate host is required. Human acquired infection by ingestion of the embryonic egg attached to their nails during stretching of the perineal area. The eggshell are dissolved by digestive juice and the larvae escape in the small intestine. Then they migrate to the cecum and vermiform appendix, where they develop to adult worm within 50 to 30 days of infection. Next, female worm may produce a pheromone to attract male, and the male worm coil around female worm with its curved area over the female genital pore. Male use spiculars to hold female during copulation. The male dies immediately after fertilized with the female. The gravid female then migrate to the perineal skin at night, stimulated by a drop in body temperature of the host. It will only lay egg on the perineum because air act as stimulant for laying egg. The injection of egg is so forceful that the egg can be spread out over the perineal area. After oviposition, the female often dies. Egg trapped in perineal fold may hatch out the larva and may enter intestine directly via the anus. This process is known as retroinfection. Occasionally, larva may enter the vulva and infect the vagina of women. Will life cycle complete within 2 to 30 weeks. Mode of transmission. Infected human beings are the only source of infection. No extra human reservoir is known. It is primarily a disease of children, and intra-family transmission is very common. The different modes of transmission are ingestion of egg due to contaminated fingers, commonly seen in children, handling contaminated nightclothes of the children, inhalation of airborne egg present in the dust. Retroinfection occasionally seen mainly in adults. Next, we move to pathogenesis. We have two types of pathogenesis, which is pathogenicity of egg and pathogenicity of adult worms. For pathogenicity of egg, significant pathological lesions are caused by the egg. The egg adhere well on surface on the skin and biting natural perineal of perineal itching. For pathogenicity of adult worm attached to the mucosa and feed on intestinal content, bacteria and possibly epithelial cell, causing minute ulceration which may lead to mild catarrhal inflammation with diarrhea, esophageal.
Next off, let's talk about the epidemiology and risk factors of Enterobius vermicularis. In Malaysia, studies have shown significant difference in the prevalence between rural and urban settings. In a study conducted in Beris Lalang, Kelantan, 79 children aged 7 to 9 were examined. The overall prevalence of intestinal helminth infection was 37%. Especially for Enterobius vermicularis, only one child was tested positive, giving a prevalence around 1.3%. In contrast, an urban study in Kuala Lumpur involving 136 children from nurseries and kindergartens showed a much higher prevalence, which is 12.5%. Interestingly, the infection rate increased with age, especially in children aged 5 to 6 years old. Globally, Enterobius vermicularis remains a major public health concern. According to the World Health Organization, prevalence among children ranges from 4% to 28%. It is estimated that around 200 million people are infected worldwide. Children aged 5 to 10 years old account for more than 30% of these infections, highlighting the global burden on school-aged children. Now, let's look at who are prone to these infections. Children aged 5 to 10 are the most infected due to the frequent hand-to-mouth activities and underdeveloped hygiene practices. Preschool and school-going children in group settings, like daycare centers or schools, are at high risk due to close contact. People in crowded living environments, such as dormitories or large households, also face higher exposure. Additionally, family members of an infected individual are high risk also because of the shared space and items. We already had a walkthrough on who are prone to these infections. Now let's see what are the risk factors that contribute to the spread of these infections. Firstly, poor hygiene practice, like for example, not washing hands after using the toilet or before meals. Second, poor environment sanitation. Eggs can survive on clothing, bedding and surfaces for days. Sharing personal items such as towels, toothbrushes or clothes increasing that risk as well. And habits like finger sucking or nail biting can introduce egg directly into the mouth.
Okay, moving on. How do we know if a person is affected with a pinworm? Because they often show no sign of symptoms. The only way to know is by during at night when the person is having a sign of stubborn itch near the anus. There is maybe a clear sign of someone having pinworm infection. In this slide, I will be explaining the diagnosis of collected samples of Enterobius vermicularis. Like I said, during at night, the worm tends to appear on the skin near the anus 2 to 3 hours after the person suspected of pinworm infection is asleep. The worm may also appear on clothes or bedding around this time. Then, conduct a tape test. Press the sticky side of a clear tape against the skin near the anus to collect pinworm eggs. To conduct this tape test is by first thing in the morning. It's the best time before the person washes, goes to the bathroom or gets dressed. Do this 3 mornings in a row. Put the tape in a sealed container, a zipping bag or provided specimen container for transport. If a glass slide is provided, apply the tape to the clear part of the slide and place in the container. Lastly, to check if the person has a pinworm infection, eggs may appear on the tape under a microscope. Moving on to the next diagnosis of finding Enterobius vermicularis under the microscope. The eggs of Enterobius vermicularis have dimensions of 50 to 60 micrometer in length and 20 to 30 micrometer in width. They are transparent and possess an elongated to oval shape with a slight flatten on one side. Typically, these eggs are partially embroidered when they are shed from the host. Other than that, the adult male Enterobius vermicularis can reach size up to 2.5 millimeter in length and have a width of 0.1 millimeter. On the other hand, adult females are much more larger than males, measuring between 8 to 13 millimeter in length and 0.3 to 0.5 millimeter in width. Males have these blunt posterior end are equipped with single spicules, which are specialized copulative structure. In other words, used for mating. Females, on the other hand, possess a long pointed tail. Next is the advanced diagnostic technique. There are 4 diagnostic techniques to identify the Enterobius vermicularis. First thing is the PCR, which is called polymerase chain reaction. The PCR purpose is to detect the DNA of Enterobius vermicularis from a sample. So I will explain how the PCR works. Okay, a swab is taken from the perianal region where female worms lay eggs. Any Enterobius vermicularis DNA is in the sample is extracted, where the PCR machine makes millions of copies of a specific DNA sequence of the worm. This allows detection even if only a small amount of DNA is present. So if DNA is present, a positive signal confirms infection. Next is the real-time PCR, which we call qPCR. It's the same as PCR, but adds quantification. That means tells how much DNA is present. So how does the qPCR work? Is by using a fluorescent dyes during DNA application. As the DNA is copied, the dye glows. The machine tracks the glow in real time and calculates the amount of worm DNA. Next is the ELISA, or what we call enzyme-linked immunosorbent assay. The purpose of it is to detect antibody or antigens related to Enterobius vermicularis in the blood. The way it works is the blood sample is taken from the patient. The sample is placed in a well coated with the parasite antigens. If the patient has been infected, their antibodies will bind to the antigen. A special enzyme plus the color reaction is added, making, if the binding occur, the well change color. The color intensity shows the antibody level. Lastly is the endoscopic or colonoscopic exam. It is the direct ablation of worms in the colon or the rectum, where you just do a surgery and the doctor can see there is a worm that can be seen on the anus. That's it for me.
Let's take a look at the conventional diagnostic method used for Enterobius vermicularis, particularly the spot flap swab method and split microscopy. Starting with the cost, these methods are widely accessible and reasonable price, making them suitable for use in school, clinics, and low resource setting. They are particularly effective when the worm burden is high, increasing the chance of tick detection. The techniques are simple to execute and they are also non-invasive, which is ideal especially for screening young children.
However, this method will come with several limitations. First, they have limited sensitivity, especially in cases where egg production is low or symptoms are absent. To improve accuracy, sample must be collected early in the morning before bathing or using the toilet, which can be inconvenient. They may also miss intermittent egg shielding, so metabolized samples across different days are often needed. Lastly, the effectiveness of this method can be user dependent, requiring proper technique in both clothing and viewing sample under a microscope. On this slide, we compare the conventional method with advanced diagnostic technique for detecting Enterobius vermicularis. Let's start with the sample type. Conventional method mostly rely on the preanal tape, while advanced method can use tape, swabs, or stool sample, offering more flexibility. In terms of cost, conventional method are inexpensive, making them ideal for mass screenings. Advanced technique like a PCR and LMP, however, tend to be moderate to high in cost due to reagents and equipment. Next, the time required. Conventional method can give result within minutes, while PCR takes 2 to 3 hours and LMP is faster at under 1 hour. Ease of use is a major advantage of conventional method. They are very easy and require minimal training. In contrast, advanced method require technical skill to perform and interpret. When it comes to equipment, conventional methods need only tape and a microscope, while advanced diagnostic require a thermocycler and proper lab setup, or at least a simple heating device for LMP. Finally, and most importantly, is the ability to detect early infection. Here's what advanced techniques stand out. They can detect early stage infection, even in asymptomatic patients, while conventional method often miss these cases. Enterobius vermicularis is highly contagious parasitic infection, especially in common young children, often spreading rapidly in group cities like school and daycares. In conclusion, the Scotch Lab test continues to be the gold standard for diagnosis. Its simplicity, affordability, and ease of use make it ideal for routine screening, especially in primary care and resource-limited settings. For the conclusion, in one molecular techniques such as PCR and LMP provide better sensitivity, making them valuable tools for detecting low burden for all asymptomatic infection. These are especially helpful in surveillance problem and for complementary diagnosis. For conclusion, finally, the most effective strategy is a combined diagnostic approach using both conventional and molecular methods. This improves overall detection accuracy, enhance infection control, and support more targeted prevention efforts.